This particular part is going to talk about the food micro lab. It basically goes back to how we work the lab. The lab is looking at food poisoning as a method to be able to understand what microorganisms do to the human digestive system when things aren't working properly. So let's take a look. It says our scenario for this sample case study, and they all start out with the same scenario. A local church group held a church picnic at 1 p.m. Sunday, May 30th. Approximately 30 people complained of gastrointestinal GI symptoms, some severe enough to visit the emergency room of the local hospital. From the information gathered from those with GI complaints, the hospital epidemiologist listed the common symptoms and average time of onset and the possible foods that might have been contaminated. Your job is to determine the causal agent from the data given. We're going to start out with sample case number B50. B50 says the suspected foods are pinto beans and sausage. The symptoms are chills, diarrhea, and cramps. Diarrhea is going to indicate lower GI. Time of onset of symptoms is 1 a.m. Monday, May 31st. We started 1 p.m. Sunday. We're at 1 a.m. Monday. That means we've got 12 hours. What they came up with is a gram-positive spore-forming bacillus as our possibility. Now, what do they do next? Procedure. Determine time of symptom development. 1 a.m. Monday minus 1 p.m. Sunday equals 12 hours. Next, determine possible bacteria based on food. We had pinto beans, and pinto beans gives us a possibility of Clostridium perfringens or Bacillus cereus. And it said sausage. Look up under meat product. We've got Salmonella, Clostridium perfringens, Staph aureus, Campylobacter, Yersinia enterocolitica. Those are the common things that you would find in sausage. What we then do is we pull all of these together. We could talk about Clostridium perfringens, Bacillus cereus, Salmonella, Staph, Campylobacter, Yersinia. The next thing we want to do is think about which of these can we eliminate. We can drop out Salmonella. Why? Because it's gram-negative. We can drop out Staph aureus because it's not a bacillus. Remember, we said it was gram-positive spore-forming bacillus. We can drop out Yersinia enterocolitica because it's gram-negative. And that kind of leaves us with 3. I'm not real sure what Campylobacter jejunii is, but the other 2 are spore-forming bacilli. That much I do know. We then look at possible bacteria based on symptoms. The symptoms were chills, diarrhea, cramps. That falls under lower GI tract. Our time frame was 12 hours. We look at the table and see what it says under lower GI tracts. Lower GI tract, abdominal cramps, diarrhea are the signs and symptoms occurring first or predominating. The first one in there is we're looking at 8 to 22 hours. The mean is 10 to 12. That fits. Abdominal cramps and diarrhea, that fits. It says what you want to analyze is stool. So you're going to take a rectal stool swab, and we're going to let them teach you how to do that in nursing because we are not doing that in our laboratory. Out of this, we get the possibility of Clostridium perfringens, Bacillus cereus, Enterococcus faecalis, and Enterococcus faecium. We can take out the Enterococcus because it's cocci, which leaves us with 2 possibilities, Clostridium perfringens and Bacillus cereus. We got to be able to differentiate between them. So what we do is we start comparing where do they have similarities, where do they have differences. Clostridium perfringens, Bacillus cereus will both ferment glucose and give you acid. They don't work on mannitol. Look at that, lactose. It says Clostridium perfringens ferments lactose where Bacillus cereus does not. So what you would do is you inoculate a lactose broth with this and see what develops. Now, that's our result on the lactose broth. That is a positive result with the production of gas. Therefore, what we've got is Clostridium perfringens as our suspected organism. Now, there are a lot of different scenarios we could do like this and a lot of different biochemical tests. This is kind of one of the easy ones. It gives you a feel on how we can go down and we could differentiate between things based on symptomology and the way we look at them. So, food micro looked at through symptomology in food poisoning.